السلام عليكم أعزائي طلبة كلية الطب المرحلة الثالثة. أنا دكتور وليد خالد سعدون، تدريسي في فرع الأحياء المجهرية في كلية الطب، واليوم إن شاء الله راح أقدم لكم المختبر العملي الأول لمادة الأحياء المجهرية. Our lab today includes three important subjects. First of all is the microscope. Second, how to prepare a fixed smear from fluid material. And third is how to prepare a fixed smear from solid material. But before starting our lab, there are specific instructions and safety rules that you have to follow while working in microbiology lab. These safety rules include the following. First of all, and the most important, is to wear a lab coat in the lab. Every student has to wear a lab coat when coming to microbiology lab, and also the gloves. Second, eating, drinking, and smoking are forbidden in microbiology lab because the environment is full of contamination, so you have not to eat, drink, or smoke inside our lab. 3. You have to wash your hands with soap and water before and after lab. Also, you have to sterilize the lab bench when you work before and after working. 5. You have to dispose of all contaminated materials in specific containers. You will see a specific containers that we will put on the table, so you have to dispose of all these contaminated materials in these containers. And 6 is that you have to sterilize a bacterial loop before and after use. This is a bacterial loop, which is an instrument that is used to transfer the specimen from one place to another. It has a circular end. This is the circular end by which we catch the specimen and transfer it from one place to another. Sterilization of the bacterial loop is done by put it over the flame. This is a benzene burner, and we sterilize the bacterial loop over the flame. By this way, we catch it by 45 degrees and put it over the flame until it becomes red in color. By this way, we sterilize the bacterial loop, and we have to sterilize it before and after use. So these are the general instructions that you have to follow while working in microbiology lab. Now we will pass to our lab today. And as I said, first of all, we will start with the microscope. Our department is microbiology. So we are dealing with microorganisms, and in order to see these microorganisms, we have to use a microscope. And this is a basic light microscope. Each microscope consists of two types of lens. These are the ocular lens. This is the ocular lens, which is the part through which the person will look. The ocular lens, as you see here, will magnify the object by 10 times. And second type of lens is the objective lenses. These are the objective lenses. Several objective lenses are carried on a turret, allowing rapid changing of these objective lens. And each objective lens have a specific magnifying power that is written on it. So you can read here, for example, for the low power lens, it will magnify the object by 10 times. And also for high power lens, for oil immersion lens, and for the scanning lens. And we will discuss this later on. These are the objective lenses. Also, we have the eyepiece tube. This is the eyepiece tube that holds the ocular and the objective lenses together. We have arm. This is the arm, which is the part through which we catch and carry the microscope from one place to another. This is the arm. And we have the stage. This is the stage when we put our slide. We can move this stage forward and backward. Also, we can move it to the right, to the left, and we can move it upward and downward by these two important knobs. These are the coarse adjustment knob. This is the coarse adjustment knob. It will be inward, and outward we have a fine adjustment knob. Now the coarse adjustment knob will move the stage coarsely. As you see, the movement upward and downward can be seen by the naked eye, while the fine adjustment knob will move it finely. Even it cannot be seen by the naked eye, but we can see the effect of using the fine adjustment knob when we are looking through the ocular lens. When we are looking through the ocular lens and move the fine adjustment knob, we will see the effect on the slide. So this is about the coarse and fine adjustment knob. Also, we have the power. From here, we switch the microscope on, and this is the illuminator that gives the light. We can decrease or increase the amount of light by this button, and also we can control the amount of light by the condenser. This is the condenser. We can decrease or increase the amount of light that strike the backward of the slide. Now, most microbiological specimens are placed on the slide that is placed on the stage. So these are the parts of the microscope. Now, how to use the microscope? First of all, before starting the usage of the microscope, we have to clean the lens, the objective lens. We clean it by specific paper which is called as the lens paper. These are the lens paper that is used to clean the objective lenses. Now we can put a drop of xylol. This is xylol, which is alcohol. We can put a drop of it on the lens paper, and we sterilize, we clean the lens by this way. Sterilize, clean the lens before use. Okay? This is the first point. Now, second, we will bring our slide and put it on the stage. Set the microscope on the scanning lens first. The scanning lens, it is the lens with the red line. It will magnify the object by 4 times. As you see here, 4 times magnify the object. Now this, the number, the 4, we will multiply it by the 10 of the ocular lens. Ocular lens will magnify the object by 10 times, and we will multiply this number by the magnifying power of the objective lens, which is 4. So using the scanning lens will magnify the object by 40 times. 4 by 10. Now we put the scanning lens and we are looking through the ocular lens and move the stage upward and downward by the coarse adjustment knob. We move it until we have the best view in the ocular lens. When we have the best view by the scanning lens, we will change to the second objective lens, which is the low power lens, yellow line lens. It will magnify the object by 10 times. We will multiply this number 10 by 10 of the ocular lens. So using the low power lens will magnify the object by 100 times. Again, we will focus using the coarse adjustment and the fine adjustment until we have the best view. After that, after having the best view, we will change to the third objective lens, which is the high power lens, the blue line lens. It will magnify the object by 40 times. Multiply this number by 10, so it will magnify the object by 400 times. Here, very important is that we use only the fine adjustment knob with the high power lens and oil immersion lens. It is forbidden to use the coarse adjustment knob with these two lenses. Otherwise, the lens and the slide will be broken. So only we use the fine adjustment knob with the high power lens. Again, we are looking through the ocular lens and use the fine adjustment knob until we take the best view. Then we will change to the oil immersion lens with white line. It will magnify the object by 100 times by 10, so it will magnify the object by 1000 times. Now the oil immersion lens is very important in microbiology. How to use it? From its name, it is oil immersion lens, so it needs oil in order to work. So we will switch to the halfway between the high power and the oil immersion lens, and we will place 1 drop of oil over the slide. This is the oil. We put 1 drop over the slide, and then we will bring the oil immersion lens to the drop of oil. By this way, the oil immersion lens will work. Again, we will look through the ocular lens and we use only the fine adjustment knob. As with high power lens, it is forbidden to use the coarse adjustment knob. We use the fine adjustment knob until we take the best view of the slide. By this way, you will reach the best view of the slide. So briefly, first of all, we will start with the scanning lens and we use the coarse adjustment knob or the fine adjustment knob until we have the best view. Change to the low power lens. Again, we use the coarse and fine adjustment knob, and until we have the best view, we will change to the high power lens. Here we use only the fine adjustment knob. Until we have a best view, we change to the halfway between the high power and oil immersion lens. Place a drop of oil on the slide and bring the oil immersion lens to this drop of oil, and we will focus using only the fine adjustment knob. This is all about the first part of our lab today, which is how to use the microscope. Now we will pass to the second part of our lab today, which is the preparation of fixed smear from fluid material. Our duty in microbiology is to find the causative microorganism that cause the infection in order to give the suitable treatment for this microorganism. So how can we find this microorganism? We can find it by prepare a smear from the specimen and put it on the slide and then stain the slide and see this slide and the microorganism under the microscope. So how to prepare a fixed smear from fluid material? Fluid material such as urine, sputum, pus, urinated exudate, or from broth culture. This is a culture which is the nutrient medium that is used for the bacteria to grow inside it. Now, this medium, this is the fluid medium, inside it there is growing bacteria. So, how to prepare a fixed smear from this fluid material? Now, what are the requirements for this? We need a clean slide, a permanent maker, bacterial loop, benzene burner, lighter, and the broth culture, which is the medium from which we will prepare the fixed smear. The procedure should be fully sterilized in order to prevent contamination from other bacteria. Now, first of all, we will take the clean slide and we make a label on the lower surface of the slide. This is the upper surface when we will work, and we make a label in the lower surface. Because later on we will make washing of the upper surface of the slide by the water. So in order for the label to stay, we make the label from the lower surface of the slide. Now, second, we light the benzene burner. We take the loop, the bacterial loop, and we take the fluid medium from which we will prepare the smear. First of all, we open the tube, sterilize it, sterilize the bacterial loop, and we take loopful from the fluid material. Now the circular end catch one loopful of specimen. Again, we sterilize the opening of the tube and close it and spread the loopful of the specimen on the center of the clean slide. Spread it by 1 to 2 centimeters to form a film of the smear. And then we will re-sterilize the bacterial loop. Now we will allow the film to dry by air. Now there is fluid on the slide. We have to wait to dry it by air until it becomes dry. Here, as you see, there is a fluid. We will wait until there is complete dryness of this fluid. So I previously prepared a slide. So we will allow the slide to dry by air without heating first until there is complete dryness. Now you see here there is complete drying of the film. After that we will fix the slide. Fixation is done by passing the slide 3 to 4 times over the benzene flame. Now the smear is fixed. And if we see this slide under the microscope, the bacteria will be colorless. So in order to view them, we have to stain the smear. And this is the subject of our second lab, which is the staining, the bacterial staining. Today, you have only to learn how to prepare a fixed smear. What are the aims of fixation? Why we fix the slide, the smear on the slide? There are 4 causes. First of all is to kill the microorganism. By fixation, the microorganism will be killed to prevent contamination later on. Second, to make the microorganism stuck to the surface of the slide. 3, to prevent the microorganism from going autolytic changes and make the microorganism more permeable to the stain that we will use it later on. By this way, now the slide will be ready to be stained. This is the preparation of fixed smear from fluid material. Now we will pass to the last part of our lab, which is the preparation of fixed smear from solid material. Solid material such as colonies of bacteria on a nutrient agar. This is a nutrient agar, and these are colonies of the bacteria that grow on it. So how to prepare a smear from one of these colonies on a slide. Again, the requirements include a clean slide that is labeled from the lower surface, bacterial loop, the benzene burner, and also we will need a clean water. Tap water can be used, and the nutrient agar that contain the colonies from which we will prepare the fixed smear. First of all, we sterilize the bacterial loop in benzene flame until it becomes red in color, and we place a loopful of clean water on the center of a slide. This is water. We take one loopful. This is one loopful, and put it on the center of the slide. Because the material is solid, so we have to use water in order to emulsify the solid material. Now again, sterilize the bacterial loop, and we take the nutrient agar, open it, cool the loop on the gel. I find an area that is empty, there is no growth, and cool the loop. And by the circular end, I will take small portion of the colony. Small portion is enough. You have not to take the full colony, only small portion. And I'll bring it to the drop of water and mix it with the drop of water on the center of a slide to about 2 to 3 centimeters to form a thick smear. And again, I will sterilize the bacterial loop on the flame. And then again, as with solid material, I allow the film to dry by air without heating. I allow it to dry by air without heating. And then after complete drying, again, I will make a fixation. As I mentioned with the solid material, I make a fixation of the smear after drying by passing it 3 times to 4 times over the flame. So the smear is now ready to be stained and to be examined under the microscope. And in the next lab, inshallah, the second lab, I will learn you how to stain this smear in order to view the slide under the microscope. That is all about our lab today. Briefly, you learn how to use the microscope, starting from the scanning lens to the lower power lens using the coarse adjustment, then to the high power lens using the fine adjustment, and then switch to the halfway between the high power and oil immersion lens, place a drop of oil and bring the oil immersion lens to the drop and use the fine adjustment knob. The second part was about preparation of fixed smear from fluid material. We place a specimen in the center of the slide, spread it, allow it to dry by air, first without heating. After complete drying, we make a fixation. And the third part was preparation of fixed smear from solid material. Again, we use 1 drop of water on the slide, then bring a small portion of the colony to this drop, separate it on the center, allow the slide to dry by air without heating, and then we fix it, and then it will be ready for the staining to be viewed under the microscope. This is all about our lab today. Thank you very much.